Yes, so in the last class we have discussed about up to the prophase, metaphase, and prophase, metaphase, anaphase we discussed, and the telophase was remaining in the last portion, right? So I discussed about that. So telophase, chromosome was condensing in the prophase. So
Mhm.
all the thing which happen in the prophase, so it is now become reversed. So finally, chromosome have reached their poles like this. Okay,
Mhm.
chromosomes have reached the poles, opposite poles like this. Okay, and now it is going to be separated from the middle, right? So we can say
Mhm.
chromosomes have reached their pole, reached their poles.
Chromosomes.
Yes,
Okay.
reach their poles. Now this is the pole. Chromosome finally, and they start decondensing. Chromosomes start decondensing,
I
or
got
we can
everything.
say,
Okay.
yeah, we can say decondensation, and become become
Finish everything, sir. I got all the
thin.
notes down.
Yeah, become thin and long. Okay,
Mhm.
we can say the thing, all the things like the nuclear membrane,
Mhm.
nucleolus.
Reappear.
Yes,
Decondensation of chromosomes. Chromosomes become big again, thin and long. Reverse effect of prophase. Chromosomes reach the pole. Their pole.
Golgi apparatus. Okay, Golgi complexes we can also say, endoplasmic reticulum. All these things disappeared in the prophase, so that's why they are coming back again. Endoplasmic reticulum. Okay, they also repaired because they were disappeared in the prophase. So of course, in the last phase, they are going to be reappeared. Okay, so it means things are coming back to the normal, and they are going
Yeah.
to divide into the two.
Mhm.
Okay,
Yep.
so the main thing is the key feature of the telophase. Simply, we can say that chromosome are moving to the opposite poles, and they are going to be divided. And nuclear envelope, everything they are going to be
Mhm.
reappeared. So finally, it is the
Mhm.
complete division of the nucleus. So
Mhm.
nucleus would become something like this. Nucleus is completely divided, but still this is complete cell. Cytoplasm is not divided now.
Mhm.
So इसका मतलब this is the cytoplasm. So cytoplasm should be divided equally. So
Mhm.
now which division will take place? I mean, which thing will happen? Can we say cytokinesis then?
Oh, well, yeah, yeah. Cytokinesis. Yeah,
Because
yeah.
the nuclear division is known as the karyokinesis.
Mhm.
So
Mhm.
after the karyokinesis, we have to take cytokinesis.
But I have two some reason here. Two.
Yeah. So see here, there is a division of cytoplasm, okay, and what will happen if there is a animal cell, animal cell. So the cytoplasm will divide something like this.
Like
There
an
is a,
eight.
yes, something like this, a furrow like this. But if it is an animal cell, it will divide from the center. Like there will be a cell plate formation, cell plate like this, and then it will be divided into two. Okay, this is
Interesting.
the
Okay.
animal cell and this is the plant cell. This is the cytokinesis.
But PMAT happens, or PMAT happens in both animals and plant cells.
Yeah, definitely. Yes. Okay. Cytokinesis is also happening here, but the pattern is different. It is coming outside to inside, and here cytoplasm is equally dividing from inside to outside.
Right.
This is the cell plate. So the
Hmm.
process of division of cytoplasm, process of division of the cytoplasm, where the two daughter nuclei, two daughter nuclei become separated, become separated into the two daughter cells. Okay, so this is for the animal cell, this is basically for the plant cell. Got it?
Mm-hmm.
Okay, so now this was about the complete mitosis. Acha, if I ask you, can you tell me what is the significance of the mitosis? Where does it take place? Can we say it is take place in our normal cells, like my
Say
normal
it
body
again.
cells? What is the significance of the mitosis?
Um. So you have.
My normal cell, my normal body cells, like
Oh yeah, yeah.
I was baby 20 years before.
46 chromosomes are there. So 46, and then it splits into two cells that have 46 each.
So
Meiosis.
it means these are
Sorry,
my all the cells, my all the hand cells, my all the cells of the body. It has a 46 chromosome except the reproductive cells.
I can't see your video.
You can't see my video,
Mm-mm.
but why?
Try how meiosis contains mitosis.
Yes, it is almost similar, not ditto
Yeah,
same.
almost. Yeah, yeah.
So now, if I talk about the meiosis, okay. So meiosis have the two divisions, meiosis one and meiosis two. Okay,
Yeah.
meiosis 1 and meiosis 2, but the actual division is meiosis 1 where the chromosome number is reduced to half.
Mm.
Actual is what? Actual is meiosis 1, so it is also has a pmet. Okay,
Uh-huh.
so p1, you're sleeping. Okay, so
No, I'm not.
p1 is prophase 1 and it has a subphases and five subphases as I just told you, right?
Lactitine,
Prophase,
zygotine,
diactin,
pactitine,
diplotin,
diglutine, and
diagnosis.
dikinesis. Yeah.
Now I want to tell you something.
Yes, sir.
Okay. You are here because of the that fertilization event, right? If there is
Mm-hmm.
any organism, so that organism is made up of sperm and ova. Okay.
Mm-hmm.
So so just tell me one thing. People usually say, "Oh, you are look like your granny. You are look like your grandpa.
Mm-hmm.
Your eyes are look like a grandmother, but your hairs are
Mm-hmm.
grandfather." How is it possible that a single sperm is containing all those character of father and mother?
Because my father and my mother have traits or genetics of their fathers and their mothers.
One second, one second, one second. I'm not talking about right now your mother character. I'm talking about your father character. Okay,
Oh,
your
okay, okay, okay.
see,
Okay, that
no, 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 no. Wait a
yeah.
second. Wait a second. Wait a second.
Yeah.
I was talking about you, Vishnu. Okay, I was.
Oh, me.
Now, yeah. So you have a character of grandfather and a grandmother. Okay. So now tell me, how is it possible that you have a character of grandfather and grandmother even after knowing that sperm has only half set of character, like the sperm chromosome? Like your dad has a cells, right? Sexual cells. So suppose of course all organisms have, but if I talk about the sperm, so sperm has only the half set of chromosome, yes or no? Half set,
Mm-hmm.
because this is what happens in the meiosis. Okay, so
Mm-hmm.
sperm contain only half set of chromosome. So in this half set of chromosome, how your grandfather and grandmother character are coming in that you are showing the character? How?
Because of relation from my dad.
Yes, you are related with the dad, but you know it very well. Your father contained the 23 chromosomes of grandmother, and your father
Mm-hmm.
contained 23 sets of your mother. So either sperms is made up of this or this. How it is
Mm-hmm.
possible that the combined characters are coming? How? Your dad is diploid, na? This is n, this is n. So that side become diploid to n. But sperm is becoming only n. So you should either show the character of grandfather or grandmother, not the both.
Mm-hmm.
But
Mm.
how you are showing all these two character?
Because my dad is a product of his father and his mother, right?
I know. and you know it very well. Yes, of course, because 23, 23. But I'm saying that why are you showing all the character of grandfather and grandmother? Because your father just transferred the only one set. Your father is like you just made from the one sperm and one ova, or the two sperm.
One sperm and one ova.
One
Two
sperm and
sperm.
one over. Okay, yes. Okay. So if you are made up of the one sperm, so just see, you know it very well. Your dad is 2n, and when the cells undergo meiosis, so these cells, the sperm will become the n. So it means either this, see on the screen, either this come into the
Either
sperm.
grandmother or either grandfather. Yes.
So that's why it become the haploid, na. So
Mm-hmm.
so within the haploid, why two characters are coming? How is it possible that the both the characters are coming into the sperm and they are becoming an grandfather character and grandmother character and sperm is taking only n. So how is it possible both the characters are going into this n and you are showing that character?
Meiosis.
Yes, meiosis is not only reducing the chromosome number to half but also doing something. Should I tell you what? Okay, see,
Mitosis. Wait, what? That made no
no,
sense.
no, 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 not mitosis.
That
I'm
didn't
going
make
to
any
tell.
sense. What the hell?
Okay, so what is happening in the father? Okay, what is happening? What happened actually in the father? See, suppose father has a. I'm talking you in terms of you. Okay, so father has a grandfather chromosome and a grandmother chromosome, right? Your grandmother, grandfather, and we can say father or mother of your father, isn't it? So see, this is the granny chromosome, and suppose this is the grandfather chromosome. So you know it very well within the when the meiosis happens. Okay, so it is same like a 23 pair of chromosome up to 23. I'm just showing you the one. So either this will go into the sperm or this will go into the sperm, right? So if this will go into the sperm, because only half is it is diploid 2n, your dad contain all these two, but when it's about this sperm, so only either this will go or this will go, right? So it means if this will go, so that baby will look like granny, grandmother. But if this will go into the sperm, it will show like grandfather. But the question is how it is showing the both the character, because if this sperm is going, so how this sperm baby is showing the character of grandfather? So it means this can only be happen when they have already combined with each other. See now what happened actually. See. Suppose this is grandfather chromosome. So before they enter into the sperm, okay, before they enter into the sperm, they have joined together. And this is known as the crossing over. Maybe they just come closer. They just come closer like this. Can you just see that? And they are exchanging their material. These chromosomes are exchanging their material, and after this is known as the crossing over. Okay, recombination, you can say recombination means they are exchanging their material, and then
Hmm.
what will happen? So it will become something like this. I will show you, don't worry.
The rest is black, maybe. What? Yeah. That's what I thought.
Now tell me, you know that sperm will have only either of the chromosome, right? Either this or this. So if the now the sperm, okay, the sperm containing this chromosome. So this chromosome have grandfather character or granny character.
Both.
Yeah, so that's why you have both the character. Got it now? So
Hmm.
it means before they enter into the sperm, so
They had to cross over.
yes, they have done this process crossing over, and they have exchanged their material, isn't it?
Hmm.
So
Yeah,
it is
I
not
see.
compulsory. Like maybe they haven't done that. They haven't done that this process crossing over. So suppose if you see a baby and that baby is only showing the character of grandfather,
Also,
no,
there's
not a
four cases. There's four cases.
four cases.
Yeah. Because you have this one. Wait, go back, go back.
Don't you
You.
think this one and this one is kind of same?
I
I
mean,
know.
not really.
Yeah, yeah, I know that you are saying that it is less character, it has a high character, right?
Is it even split like 50-50?
Yeah, you can say that. Yes, yes, yes. Yes.
Okay, so then
You
there's
can't
three
trace.
cases.
Yeah, yeah, yeah. There is no not 100% proof how much character you are going to take. Okay. So because anything can happen, we are not sure it is a 50-50%. We are not sure it is a 60-40%. So whenever this happens, so we consider this chromosome and this chromosome as same. Okay, because this
Hmm.
has a both the character, this also has a both character. Okay.
Okay, so there's three.
Yes, one is granny, one is grandpa, and one is combined. Right?
Okay, that's cool.
Got it now?
Yeah.
So it means if a baby is showing grandfather, grandmother of that side, grandfather, grandmother, nanny, okay, mom side, all four character. It means mother chromosome also has done this
Yeah.
before they become the ova, and sperm, like your dad also has done this before they form the sperm formation, right? So is that clear, everybody?
Yeah.
Okay. So this is how things happen. So so you know it very well. Now meiosis brings variation. So because of this variation will come, right? Like in a single
Yeah.
product, all the product you will see, all the things, all the new character, right? So you can see that prophase one it is. This is prophase one, right? And prophase 1 have 5 substages: lactotine, zygotine, pectine, diplotin, and the dikinesis. Okay, so now you should know I am going to teach you the meiosis part, because meiosis 1 is important, but I told you meiosis second is just like the.
Mitosis.
Yes, so obviously first should be karyokinesis or cytokinesis.
Karyokinesis.
Yes, so let's
The the
start
the
with.
the, the nuclear one.
Yes. So meiosis one, karyokinesis I'm going to start. And the first one is the prophase. So prophase one is divided into how many parts?
Two.
Five. Lactotine.
Oh. Shit. Okay, no, you're right. It's yeah, those. But like
Okay.
prophase is divided into two.
Yeah. No, everyone knows, wait a second. No, that was meiosis. Meiosis divided into two. Meiosis one, meiosis two.
No, but no. But then what is prophase one and prophase two?
Prophase 1, prophase 2, it is a part of the meiosis 1 and meiosis 2. So as I can say that anaphase 1, anaphase second, metaphase first, metaphase second,
That's
telophase
so
1.
confusing, bro. What the heck?
Okay. See. Don't
Okay, then it's
agree.
five.
It
Yeah,
is it is
the five
meiosis.
things.
Yeah, meiosis and meiosis 1, meiosis 2. Okay. So meiosis 1 has all the prophase, metaphase, anaphase, telophase. All these are one. It is a part of meiosis 1. And it is also prophase, metaphase, second. It is a part of second. Okay. So but prophase
I
1
see.
contain only the 5 substages. So let's start with the lactotine.
Wait, question. What is lactine an enzyme or like what are they?
Actually, lactotine name is given on the like condition when the chromatin become a very short type. They look like a very fine thread like structure. This is called lactotine. So that's why I
The
can say
process.
yes.
Okay, so chromatin becomes small and
I'm writing here.
thin.
So and coiling of condensation and coiling
So decrepit, no, not decrepit. No.
coiling of chromatin fibers. Okay. So whatever the chromatin fibers they have, so it is start condensing and coiling are going to take place, and it lead to the formation of distinct chromosome, distinct chromosomes. Okay, and they look something look like a fine single thread. Okay, like this fine single threads. This happens in the lab protein, and they contract at the highest level, you can say. So finally it will look like a they contract and become thicker. Okay, so contract and
Mm-hmm.
they thicker. So this is the prophase one. Okay, now if I talk about the so this is only these things are only happening in the lab protein. Now I showed you these thing, you saw, yeah, this one. So this is
Mm-hmm.
known as the homologous chromosome. Like right now, if I talk about myself, so my body has dad character and the mom character, right?
Mm-hmm.
So it means this is the homologous chromosome. So before I release these sperms, okay, so I have the homologous chromosome, and there should be recombination, there should be crossing over, right?
Mm.
Oh,
Uh,
there is a so much cold, and you are like fan is running. My
it's
God.
always running.
Oh, so you don't feel any cold?
Oh no, I'm wearing like sweatpants.
Ah,
I'm wearing sweatpants, like thick stuff. I have like a bunch of blankets here too.
okay, cold man. Okay. So now we have the zygote and the homologous chromosome start. Yes, you were saying something before I asked something. Yes, you were saying something. Yeah. No.
Oh,
Okay,
I was
okay,
just gonna say like nothing. I didn't have any questions.
okay. So homologous chromosome means, okay, can you tell me what is homologous chromosome?
A homologous chromosome is when
I showed you
like
just.
yeah, the crossovers, right?
Yes. So in your body, you have a one set from father directly and
Mm-hmm.
one set from mother. That's why you are 46.
Yeah.
So all these two set, like father and mother, it is known as homologous chromosome. So pairing, crossing over. Suppose if you're going to release this sperm. So if you're going to release this sperm, so before that, in your body, crossing over will take place. So that
Mm-hmm.
crossing over will take place in dad to dad, mother to mother, or dad to mother.
Wait, say it again.
When you're going to release this sperm, so crossing over will take place in your body.
Mm-hmm.
Okay, so that crossing over will take place to your dad chromosome to dad chromosome, mother chromosome to mother chromosome, or dad chromosome to mother chromosome.
Dad to mom.
Yes, so that chromosome is going to cross over with mother chromosome, right? So it will form a combined chromosome, right?
Yeah.
So that combined chromosome, suppose if you have a baby in future, so that will show only one character or two character, both the character, right? Okay. So we can say that in the this is the lactotin, right? Prophase one. This is the zygotin, second one. So always remember, they will help in the formation of. So when they come closer, okay, something when they are something closer. So this is known as this process is also known as the synapses. Remember, and this is known as
Mm-hmm.
synaptonemal complex. Remember. So the formation of synaptonemal complex, synaptonemal. So the in the zygote formation of synaptonemal complex take place, okay, and these are known as the bivalents. These are known as I mean they look like their name is given to you. This is known as bivalents. Okay, see what I'm saying. Whenever the two chromosome, dad and mother, they are coming closer in your body before releasing this sperm. Okay, so this is known as synaptonemal complex, and a name is given to that. This is also known as bivalence. Okay, so this is only
Mm-hmm.
thing only happen in the zygote, but the main part now, main part happen in the middle part, which is the pachytene.
What happens here?
So now you saw that process, they are coming
Mm-hmm.
closer completely. So
Mm-hmm.
the two sister chromatid of each chromosome are completely visible, and they are now look like a tetrad. Okay, tetrad, because there are one, two, three, four. One, two, three,
Tetrad.
four. Yes, so look like a tetrad. Okay,
Tetrad. Okay.
okay. So firstly, they were bivalents. Okay, bivalents separately, bivalent, and
Mm.
now bivalent become tetrad. Okay, total four. So two sister chromatids.
Becomes tetrad.
Yeah, two sister chromatid of each chromosome, right?
Mm-hmm.
So they will form what? Tetrad. Or we can say this quite visible. So now finally here recombination is taking place, right? Here
Mm-hmm.
a recombination is taking place. So recombination is happening, and this is also known as the crossing over. So crossing over is happening here in this pectine. Okay, and
Mm.
there is an enzyme like this process when the two chromosomes are coming closer and they are exchanging their material. So there is an enzyme which is called recombinase. So remember, crossing over is catalyzed by enzyme recombinase. Okay, so why
Recombinase
recombinase name is given to that?
is
Because the
recombining everything.
yes, recombining, there is a mixing of both these, right? So mixing of these things are happening. That's why the enzyme name is recombinase.
Yes. Okay. Yeah,
Okay,
I get it.
lipoprotein, glycogen, pectin. Okay, so now they have exchanged their material. Now things have exchanged. So now they are going to be separated, or they are remaining together.
Hmm. What?
Now they have exchanged their material right here. So now
Uh-huh.
once they have exchanged their material, so they are going to be separated, or they remain close.
I mean, they have to like come
They
apart.
have
I mean, they have.
exchanged the material. So if they have exchanged the material, so they will come back again, or they will remain like this.
I mean, I think they have to come back at some point, right?
Yeah, told you. Can you just see that? When they come closer, so after the closer, they just separated, na?
They have to come back at some point.
Yes, yeah. But the chromosome was the same. This one and this one. No.
No, they after they cross over, they're two different ones, right?
Yes, it is a kind of mixed chromosomes, right?
Yeah, it's like a mix and
So,
match.
so it means one chromosome contain both the character now.
Yeah.
So we can say that it is a dilution of synaptonymal complex. Dilution, like whatever you just saw. So it is dilution of these things take place. So the dilution of synaptonymal.
Okay, so diptotine, diplotine,
And can we say that these homologous chromosomes start separating now because they have exchanged the
dissolution
material?
of what?
Homo
Complement?
synaptonymal complex. You remember this was formed in the second stage. C, zygotine, synaptonymal complex. They are coming closer, so they form a complex which is called synaptonymal complex. Okay,
Wait,
so the
go back. Just one.
sure, sure. So the homologous chromosomes start separating from the each other. This is called the diplotin. Okay,
Hmm.
okay. Actually, these chromosomes are come close to every path. They are trying to come closer, okay, in the packaging. But now they are going to be separated, okay. Chromosomes start separating from the each other. Okay, so now it will look like something like this. See. So it will something like this, like they have almost separated, but not the ends. Can you just see the spotlight?
Mm-hmm.
Like they are completely separated, but not this side. This side haven't separated yet. Like see, they were
Mm-hmm.
very close to each other. Now they are separating, but still this is attached. Okay. So once this is attached, so this is known as the what? So this is known as the
Synapse.
chiasmata. Yeah, synaptonymal complex, you can say it is going to be like dissolved, but this complete name, when only the you see crossing over this side only, so this is known as chiasmata.
Oh,
Chiasmata.
okay.
Okay, so
The
where will you,
process.
see the chiasmata? You will see only the
The
diplotin.
diplotine.
Yes, and then in the next last stage, they are also going to be separated, right? So the last stage is dikinesis.
So the process when homologous chromosomes like divide or like drift apart.
Yes. So finally, chiasmata will also be slip off, or they will remain attached.
Slip off.
Yes. So the chiasmata move to the tip. I mean, tip means terminalization will happen, and they slip off.
Wait. Okay, the chiamata is when they drift apart, right? The homologous
No,
chromo.
this complete structure is known as chiasmata.
Oh,
Okay, this
okay.
complete the structure which is looking like something like this. They are attaching at the tips. Okay. So remember.
Okay, I
Okay.
see.
So
So
finally, it
when
will.
so the chromosomes is it any chromosome or is it the crossover chromosomes?
What? What?
So is the chiamata only the regular chromosomes or is it only the homologous chromosomes?
Only the homologous chromosomes.
Okay.
Only when they are just crossing over. Okay.
Okay. I have to. That was my question.
Yeah. Do you have one more question?
So only when the homologous chromosomes are attached at the tip.
Because only the chromosomes, only the homologous chromosome are doing this process, na? Not every chromosomes. Homologous means one chromosome of mother, one chromosome is of father. Mother of
How
father.
do you
So
spell? How do you spell chiamata? Whatever that is.
I wrote
Um,
here. Can you just see that?
I can't. Okay, there.
Chaos matter. So actually, question will be asked: where will you see the chaos matter? It is diplotin. Where will you see
Homologous. Oh,
the
okay.
crossing over main recombination is taking place? Pachythene. Where will you see the synaptonymal complex? Zygote. Okay.
Hmm.
And finally, it is just slip off. So all these are what? So this is the leptin, zygote, pachythene, diplotin, dikinesis we have done. So can we say we have done the prophase one here?
This is like these five things are mainly just regarding chromosomes.
Yes, and it is only happening in the prophase one.
Oh yeah, yeah,
Okay.
that's facts.
Okay, and yes, in the, because it is the last phase of prophase, so prophase thing also happen here. You remember all these like disappear, nucleus, nuclear membrane disappear. So
They
this is the,
reappear, right?
yeah, no.
They
This is the,
disappear.
yeah, because this is the prophase one of
Yeah,
meiosis. So that's
that's what I
why.
said, right? Did I not say that? Oopsie, I meant disappear. I
Okay,
mean disappear.
okay. So have you note down these things
Yeah, yeah, I did.
now? So after the prophase one, we have the metaphase one.
Mm-hmm.
So in the metaphase one, what is happening actually? So all the tetrad you just saw, the bivalents, the tetrad, okay, the bivalents, or you can say the tetrad, okay. So of course, all these are arranged at the equator.
Mm-hmm.
All these are arranged at the equator. I told you, like like it is the cell, and my body contain two set of chromosomes, so they are arranged at like this here at the equator. And now from this spindle will come, right? And they will attach with the kinetochore I told you, right?
Mm-hmm.
And they are trying to push it towards its side.
Mm-hmm.
Okay. So they are and finally half of the chromosome will go there and half of the chromosome will go there, and finally it is
Oh,
separated.
it would
And
divide it. Yeah, yeah.
from the 2n to nn. Okay.
Mm.
So the bivalents arrange at the equator of the spindle. So they are arranged at the rows. You can see that centromere
Oh yeah, forgot that centromere.
are aligned in rows. So this is the metaphase one. Now, if I talk about, so this is that was the prophase one, this is the metaphase one, and now we have the anaphase one.
Mm-hmm.
So in the anaphase one, the members of each or every homologous chromosome, so they are going to be separate from each other, and they start moving to the opposite pole. Am I right? So
Mm-hmm.
they are going to reach like this,
Mm-hmm.
and this one also. So they are going to reach the opposite poles. So we can say there's the members of every homologous pair of chromosomes chromosomes separate from each other and start moving to the opposite poles of this spindle, right? One is going to that side, other is going to that side. And remember, the centromeres do not split. Fine. So this is
Yep.
the anaphase one. Now, if I talk about the telophase one. So if I talk about this telophase one, so the chromosomes have reached the poles.
Mm-hmm.
So they have reached towards the poles, and we can say that the nuclear membrane forms around the chromosomes, okay, towards the each pole. So finally, it means the chromosomes are separated here, and the chromosome here. These are the separated here. Okay, so both are separated here like this. Got it?
Mm-hmm.
So this is the telophase one. See, the chromosome have reached towards the pole, and finally equal chromosome. Like firstly you had the 2n. So from the 2n, n are going to come at the pole, and the n chromosomes are going to the another pole. So
Mm.
this happens in the telophase one. Okay. So and
Mm-hmm.
finally this is completely karyokinesis.
Yeah, it's the nuclear division.
Nuclear division has done,
Yeah.
and
Now
then
it's
cytokinesis.
cytokinesis.
So you know it very well. Then cytoplasm will be divided same like as we have
Mm-hmm.
seen. So cytoplasm equally divided. So
Yeah.
I told you, when you have this cell, so these are going to form the two cell. Okay. So only after meiosis one,
Mm?
and these are going to form the two more cells only after the meiosis 2, right? This is
Yeah.
meiosis 1, this is meiosis 2. So we have done till now, but these cells are not so developed. Okay, here. So the daughter diodes are, the two cells are formed now. So we will call it as two daughter cells are formed. And these two daughter cells are known as the diodes. So they are formed. Fine, this is cytokinesis. Got it?
Yeah.
So just after the meiosis 1, it is not going to enter into the meiosis 2 because it needs some break. Like if you're
Hmm.
continuously studying, you're continuously working, so you need a short break na. Like
Hmm.
because here like there is no replication of DNA take place, remember, but it is something like the gap, interkinesis. It is a gap between the uh, like meiosis 1 and meiosis 2. Okay, see, the meiosis main purpose is to divide the chromosome, I mean to equally, see, if you
By
have
the
the total
cell, yeah.
if you have the total 46 chromosomes, so the main purpose is to divide into the equal to, right, 23 and 23.
Mhm.
So this has already done by this, yes or no?
Yeah.
So meiosis 2 also dividing.
No.
No, so if it means if further they will go into meiosis 2, so will it become 12.5?
Oh no, 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 no. It has to be the same. So that
Yes,
becomes mitosis because
yes,
it has to
from
be the same. No,
yes,
we talked about this. I think we talked about this
yeah.
last class. I remember.
Yes, exactly. So it means which one is the actual meiosis? Meiosis 1 or
Oh, the
meiosis 2?
first one. The first one.
Yes, okay. And I told you meiosis 2 is just like the mitosis, so the all the events will remain the same, right?
Yeah, PMAT.
Yes, whatever happened in the mitosis, so it will happen in the meiosis too. So can we say that what happens in the prophase second? Just tell me.
Mitosis.
Yeah, but still I'm asking you. Prophase second, can we say that all the cell membrane, all the endoplasmic reticulum, Golgi apparatus, they just disappear?
Yeah.
Okay, and we can say nucleus also disappear.
Yeah, the nuclear. Yeah, yeah, yeah.
Okay.
Nuclear membrane.
Okay. Second, we have the metaphase second. So can we say that like a spindle fiber formation are taking place and it will
Hmm.
attach to the kinetochore, right? Kinetochore. A
Yeah,
spindle will attach to the chromosome and then in the
yeah.
anaphase they are going to the other side and they are like spindle fiber will pull up, right? It will push it.
Hmm.
So finally, you will see something like this. So this will happen in both the cell we just formed in both the cells.
Yeah.
Diet. And finally, so finally, how many cells you will get after telophase? Like things will reappear, and finally it will divide it. And after the cytokinesis, so how many cell you will see? Total
Two.
two or total four?
No, four. Right. Oh no, no, no. We went over this. We went over that. It was four.
Yes. Oh, total we get four, right?
Yeah, but I okay. Technically, it's six, but it's actually four.
Technically, it's six. Why? You are also considering this, right?
Yeah, those two.
No,
Yeah,
these two
those don't
form.
count. They don't
Yeah.
count. They don't
Okay.
count.
Because they are converting into two.
Yeah,
So they are not
I.
left.
Yeah, yeah.
Fine, I
That's
got it. Technically, it is six, but actually, it is total four because two are
yeah.
converting the into the four. Okay, so and cytokinesis, you know it very well. Complete division of cytokinase, right? So with that, we have completed the this chapter, meiosis and the mitosis. So if I
Thank
ask you
God.
what is the significance of meiosis,
Meiosis is significance.
can we say this is how chromosome half
Reproductive
of,
cells.
yes, this is,
Reproductive cells are
can
produced.
we say that also this is how variation are coming, variations,
Oh
new
yeah,
new
yeah. It
character,
has a variation.
because baby has so many character of other families, different families, different characters, right? So
Hmm.
this is why how these things are happening, because of the meiosis. Okay, so
Variation.
meiosis is the one which is taking place in the reproductive cells and they are maintaining the same number of chromosome. See, in
Hmm.
humans male and female are there, right? So if male
Wait,
and
no,
female are
not
there,
the
so
same number of chromosomes. Half the number of chromosomes.
see 46, suppose if dad is giving 46, if mother is also giving 46, so baby become 92 na.
That's impossible.
Yeah, so that's why we need this division meiosis, right?
Yeah,
Because if meiosis were not there, then it would be mitosis and then chromosome keeps on increasing in the next generation, so and that is
it's
not good.
weird. You can't. It's just like
Yeah,
not impossible.
yeah, so that's why meiosis is very important for the human reproduction. Okay, for maintaining the same number of chromosome. Okay, any doubt in this
Mhm.
meiosis?
Nope.
So all the events you have to remember, okay, what happens in the which state? And cytokinesis, if I ask you, okay, there is a one more thing. The cell which enter the reproductive cell, they enter into the reproduction. Okay, so it is a meiocyte. So
Mm-hmm.
what is a meiocyte? A meiocyte is a cell which enter into the meiosis. Okay.
Mm-hmm.
So in the next class, when you come, I will ask many questions. Okay,
Mm-hmm.
on this. Is it fine to you?
Yeah, I'm good.
Okay. So do you wanna take class tomorrow?
Oh, I can't tomorrow.
Okay. Okay. Fine. After tomorrow,
Maybe can we do Thursday?
Thursday. Okay.
And then I
Your Thursday.
yeah, my Thursday.
So it is the usual. It is the usual schedule.
Yeah.
Okay.
Okay.
Then fine. Fine
No,
then.
no, no. Okay. No, I'm not done yet. Then we can, if you are free, we can also the weekend, like Saturday, Sunday.
Oh. Okay. Okay. Then we
Wait,
will
are
talk
you gonna be
about
out of town?
this in the next
Yeah, yeah,
class.
yeah. That's fine. That's fine.
Okay. So
All
this
right. Bye.
chapter
Thank
is.
you.
Bye bye. Take care.
Bye.